Clinical Chemistry, Introduction to Clinical Chemistry and Lab Safety. This is a list of the course competencies and learning objectives that we're going to discuss in this section. How do we diagnose diseases? So there's several different areas in the clinical laboratory. Clinical chemistry, which is the focus of this course, evaluate analytes from blood samples to identify which cells or organs may be injured. In molecular diagnostics, they evaluate genetic material to identify the presence of select genes or errors within a gene. In microbiology, they evaluate specimens for the presence of pathogens and attempt to isolate that pathogen to determine its identity and susceptibility to drug treatments. Although nowadays, a lot of microbiology is going toward molecular diagnostic tests. Immunology and serology use antibodies or evaluate the presence of antibodies to detect diseases. So they're going to evaluate different samples using antibodies to determine if there is a select disease or protein or antigen present in the blood, or they're going to evaluate the presence of antibodies to determine exposure to a particular disease or infectious agent. Urinalysis, they evaluate the urine for abnormal constituents to determine a cause of disease or provide further evidence of the disease. Hematology is predominantly going to evaluate the blood cells to determine whether there are abnormalities that could cause disease. And then cytology or histology, they evaluate cells or tissues for the presence of disease, predominantly cancer with cytology and histology. So all of these different areas work together to evaluate patient specimens so that we can diagnose what is actually wrong with the patient. So the clinical chemist is going to look for analytes in the blood to determine whether there's a disease state going on. So what are analytes? Analytes are the chemical substances that are being identified and measured. Analytes could include antibodies or antigens. That's predominantly going to be in immunology. Carbohydrates, lipids, proteins, including enzymes, amino acids, peptides, non-protein, nitrogen, hemoglobin, bilirubin, and hormones. Vitamins, minerals, ions, blood gases, trace or toxic elements. Drugs, including drugs of abuse and alcohol. And markers for bone, tumors, diabetes, fertility or hormones, thyroid function, cardiac, hepatitis, infectious diseases, sepsis or inflammation, and other types of markers. What are the duties of the clinical chemist? Develop and validate tests, evaluate and characterize tests, present results in an effective manner, perform algorithms and clinical practice guidelines, regulatory compliance, and research. So the clinical chemist is always thinking about how can I improve on this process. A lot of clinical chemistry today is automated. So typically, doctor orders a test, we get a blood tube down in clinical chemistry. The clinical chemist puts the blood tube on the machine or the analyzer, we like to call it, and then the analyzer will perform the various tests. The clinical chemist may or may not have to add reagents to it, but they're definitely going to have to look at the blood sample itself before they put it on the machine to determine if there's any hemolysis, lipolysis, like additional lipids in there, cloudiness, and then clots are a major factor in clinical chemistry as well. So clots could interrupt the function of the analyzers. So develop and validate tests, that's kind of more on the research side. Typically in a hospital or healthcare situation, you're not going to be developing new tests. And if you do develop new tests, those all have to be passed through the appropriate checks to make sure that they're valid. Evaluate and characterize tests. That's something that's done a lot with QC to make sure that the tests are functioning properly. Present results in an effective manner. So that's something where the clinical chemist is going to use the majority of their time. So they're going to look at the results that came from that particular sample and determine, do those results make sense? Is it time to release those results? Or are there any critical results in there that we might have to call up to the floor or call up to the treating physician? Algorithms and clinical practice guidelines are just part of the job. So that's something that you do on a day-to-day basis. You want to make sure that you're following all protocols and procedures and also guidelines for your laboratory. Regulatory compliance kind of goes along with that one. And then research. So research is kind of above and beyond the duties of a typical clinical chemist. But there's research studies that happen all the time that evaluate different concentrations of different substances in the blood and attempt to correlate it to a certain disease state. What are some ethical issues encountered in the lab? So commonly encountered situations, confidentiality. So patient confidentiality is extremely important. That's why we have HIPAA in place and all that type of stuff. Genetic information and patient's medical information is all protected and should be kept confidential. You should only be pulling up patient records when it's relevant to performing your job. You shouldn't just be looking up patients out of curiosity. Allocation of resources. So the best use of time and cost versus quality. Codes of conduct. Publishing issues. So if you do venture into research, there's fraud, plagiarism, falsification, or fabrication of data. Unfortunately, this happens a lot more than one would suspect. Even in research institutions, this is something that is an ongoing issue where publications come out because they were pressured, the doctors were pressured to publish something and they just hurry results or falsify data to get their publications moved forward. And then conflicts of interest. So sometimes conflicts of interest exist, and those are typically reported, especially if you're going to publish a research article. You're very transparent about conflicts of interest. So now that we've talked a little bit about clinical chemistry and what the typical clinical chemist does, we're going to talk a little bit more about laboratory safety. So here's some lab safety definitions that you should know. So what is a laboratory? It's a facility where the laboratory use of hazardous chemicals or biological specimens occurs. It is a workplace where relatively small quantities of hazardous chemicals or biologicals are used on a non-production basis. Biosafety. The principles and practices employed to protect laboratory personnel, students, and the environment from exposure or infection while working with living organisms, biological materials, or agents. Chemical hygiene, the principles and practices employed to protect laboratory personnel, students, and the environment from exposure to hazardous chemicals. In the clinical chemistry area in the hospital, laboratory, or clinic, biosafety and chemical hygiene are both practiced. Most of the patient specimens that are received are biohazardous materials, they're blood. And then we talk about bloodborne pathogens and all that type of stuff as we go through a little bit more of this PowerPoint here. The chemical hygiene is another topic. We do use chemicals in clinical chemistry. We add reagents, and sometimes those are done benchtop, sometimes those are done by the analyzers, but if the analyzers go down, then we end up reverting to using the benchtop addition of chemicals. Chemicals and reagents also have to be replaced on those large clinical chemistry analyzers, so that's another potential source of exposure. A health hazard, chemicals which are carcinogens, mutagens, teratogens, toxic agents, reproductive toxins, irritants, corrosives, sensitizers, hepatotoxins, nephrotoxins, neurotoxins, agents which act on the hematopoietic systems, or agents which damage the lungs, skin, eyes, or mucous membranes. Physical hazard is a chemical or material that is combustible liquid, a compressed gas, explosive, flammable, an organic peroxide, an oxidizer, pyrophoric, unstable or reactive, or water reactive. A biohazard is a biological agent or specimen that poses a risk to the health of humans, animals, or the environment. In other words, most biohazards we consider to be living organisms. However, viruses and even small infectious proteins like prions can be considered biohazardous agents. A bloodborne pathogen is an infectious microorganism in human blood that can cause disease in humans. The number one most common bloodborne pathogen encountered by laboratory personnel is actually hepatitis B. Microorganism, an organism that requires the use of a microscope to be seen. Examples include bacteria, archaea, protozoa, algae, and fungi. Acellular particles such as prions, viroids, and viruses, or the eggs of helminths, which are cellular, are also classified in the field of microbiology. Body fluid, liquids from within the human body, which are evaluated in a laboratory. Examples include saliva, semen, vaginal fluids, mucus, urine, and others. Biological agent, microorganisms and their associated toxins, which pose a human health risk. And then here's some commonly encountered abbreviations. So BBP is a bloodborne pathogen, and that's was defined in one of the previous slides. A BSC is a biosafety committee. BSC can also represent biosafety cabinet, so it's important to read the context of that. But biosafety committee is basically a committee of personnel that are designed to ensure best practices for biological safety. The biological safety committee is typically run by a biosafety officer, a BSO. And the BSO is responsible for developing and maintaining a biosafety plan, or a BSP. In addition to biosafety, we have chemical hygiene, and chemical hygiene is chemical safety. So we have a chemical hygiene officer, which oversees, develops, and ensures the accuracy of the chemical hygiene plan. And then they have a chemical safety committee that evaluates best practices for having the safe use of chemicals in a laboratory. Environmental health and safety encompasses all of the safety and environmental hazards and health issues that could be encountered within a workplace. An LSC is a laboratory safety coordinator. Laboratory safety coordinators are more commonly found on in schools, universities, that type of stuff. So those are going to be the people that oversee laboratory safety and ensure the safety of students during the laboratory experiments. MSDS is material safety data sheets that gives information of all of the different hazards associated with particular materials. OSHA, Occupational Safety and Health Administration. PEL is the permissible exposure limit. So the permissible exposure limit is how much maximum of a substance can a person be exposed to and still be safe and still not have any health hazards. Primary investigator or instructor is the PI. So that's the person who is in charge typically of a research lab or an academic lab. PPE is personal protective equipment, so that includes gloves, goggles, all that type of lab coat stuff that's going to protect you against exposure to chemical hazards or biohazards. Pathogen safety data sheets, or PSDS, give more information about different types of organisms that are used within a research lab. Sometimes it's a research lab, sometimes it's going to be a clinical lab. We order in a lot of reference strains so that we can do our use them as positive or negative controls in different tests. We can also validate test systems using those reference strains. So they can be found there and also in like classrooms. SOP is a standard operating procedure. So the Occupational Health and Safety Act, or OSHA, public law, is a federal regulation enacted by Congress in 1970. The goal is to provide all employees with a safe work environment. There are on-site inspections to ensure that there's compliance, and it's administered by state agencies or federal administration. Safety is no longer only a moral obligation, but also a federal law, and includes standards that regulate safety in many aspects of the clinical laboratory. If OSHA finds violations and those violations are not corrected, the laboratory can be shut down. So this is something that's pretty serious. The Bloodborne Pathogen Standards mandates universal precautions to prevent, evaluate, and manage exposure to bloodborne pathogens. Universal precautions presumes that all blood, tissue, and fluids are infected. So in other words, you treat every specimen as if it contains a highly deadly infectious agent. That way, If it actually does, you've already been practicing those precautions. And if it doesn't, then at least you're still safe and you're not going to be exposed to anything that is in the blood. Defines exposures and mandates the development of an exposure control plan. An exposure control plan describes what you're supposed to do after you've been exposed, and it talks about how to prevent exposure. Hazard communication standard defines hazardous chemicals and ensures employers and employees are made aware of hazards. The means of communication are through proper labeling, the development and use of safety data sheets, and employee education. Occupational exposure to hazardous chemicals in laboratories requires a chemical hygiene officer and chemical hygiene plan. So if there's ever chemicals used in a lab, which is every clinical lab, there should be an appointed chemical hygiene officer and an associated chemical hygiene plan. So for safety awareness, the employer is responsible for establishing laboratory work methods and safety policies, provide supervision and guidance to employees, provide safety information, training, PPE, and medical surveillance to employees, provide and maintain equipment and laboratory facilities that are free of recognized hazards and adequate for the tasks required, know and comply with the established laboratory safe work practices, have a positive attitude towards supervisors, coworkers, facilities, and safety training, be alert and give prompt notification of unsafe conditions or practices to the immediate supervisor and ensure that unsafe conditions and practices are corrected, and engage in the conduct of safe work practices and use of PPE or personal protective equipment. So a lot of hazards are communicated using appropriate signage and labeling. The National Fire Protection Association's Standard Hazards Identification System is a diamond-shaped color-coded symbol. There's also manufacturers' precautionary labels that have a statement of the hazard, precautionary measures, specific hazard class, first aid instructions, the storage code, safety code, and protective equipment that's needed. So every chemical manufacturer would put that on the container that they ship out their chemicals to various locations. This is the NFPA hazard sign. I have seen this come up on the qualifying exam for ASCP, so this is something that you should know what the colors mean and know what the number system is, because they can get pretty specific about this on your qualifying exam. So blue refers to a health hazard, red is a fire hazard, yellow is reactivity, white indicates other types of hazards, and then this is on a scale of 0 to 4, where 0 it's no hazard, and it kind of progressively goes up to 4, which is an extreme hazard. Some safety equipment in the laboratory. Fume hoods expel noxious and hazardous fumes from chemical reagents. They should be inspected for blockages, ventilation velocity, and they should never be operated with the sash fully open, and a maximum operating sash height should be established. So a fume hood is different from a biosafety cabinet in that a fume hood typically takes the fumes that are released from chemical reactions and dissipates them outside of the building. So it'll go through some filter systems and then it will expel the fumes out into the environment. When those fumes leave into the environment, they're diffused enough so that it's at a safe level. So if there was an animal or a person standing outside where it did vent out, it would be immediately kind of diffused out and it would be at a safe level. There is a sash, so there's a window at the front of the fume hood, and that sash can be moved up and down. Most fume hoods have a space where it's safe to operate that fume hood, and if you move it above that line, you open up the sash too much, an alarm system will go off, and it will require you to lower down that sash down to an appropriate level. Biosafety cabinets remove potentially harmful particles of infective biological specimens. They offer various levels of protection depending on the biosafety level of the specific laboratory. So there's different types of biosafety cabinets, but their goal is predominantly to filter the air and recirculate it into the room. So you're going to be using certain types of microorganisms within the biosafety cabinet those are going to produce spores or toxins or different types of particles that can be released, or the microorganisms themselves. Then the air inside of that biosafety cabinet will go through a HEPA filter system, and after the HEPA filtration, that air will recirculate into the room. Biosafety cabinets can typically be freestanding units that just require an outlet to plug into the wall because they are recirculating the air into the room after passing it through a HEPA filter. HEPA filters, if you have heard of from microbiology, do not capture viral particles or anything smaller, any smaller type of bacteria. So although they do, they are able to capture most mold spores and that type of stuff, there are things that can go through those HEPA filters. So that's just something to keep in mind. In microbiology, when we receive specimens down to the lab, we're typically going to be doing all of our plating underneath the biosafety cabinet. And we're going to open up the patient specimens and plate them and do our gram stain slides and everything underneath the biosafety cabinet. And then the biosafety cabinet is routinely cleaned, and most of them have a UV light, and UV will kill most microorganisms. It won't kill really tough resistant microorganisms, including endospores, which are a huge issue. And that's where we use disinfectants and such to kind of clean out the biosafety cabinet. Biosafety cabinet is rarely encountered in the clinical chemistry area. Chemical storage equipment. So they're safety carriers for the transport of acids, alkalis, or other solvents. Safety cabinets, safety cans, and explosion-proof refrigerators for flammable liquids. Gas cylinder supports or clamps, the use of hand carts for large tanks. Personal protective equipment, PPE, and hygiene. Safety glasses, goggles, visors, work shields to protect eyes, gloves, rubberized sleeves, full-length lab coat, proper footwear, proper respirator, so a HEPA filter respirator or an N95 mask. Hygiene and hand washing is also important. So biological safety. As I mentioned previously, the most common biohazard that you're going to be exposed to is blood in clinical chemistry. So if you spill some blood, cleanup of spills is essential. So you want to alert others in the area of the spill so they don't accidentally happen upon it and get exposed themselves. Wear appropriate protective equipment. Use mechanical devices to pick up broken glass or sharp objects. So you never want to pick up glass with your gloves or with your bare hands, especially once it's contaminated with a patient's body fluid or blood. Absorb the spill with paper towels, gauze pads, or tissue. Clean the spill site using common aqueous detergent. Disinfect the spill site using approved disinfectant or 10% bleach, using the appropriate contact time. So some people will just spray the bleach on there and immediately wipe it up. There should be some time where the bleach has an opportunity to interact with the particles to give it enough time to kind of prevent further hazard. Rinse the spill site with water and dispose of all materials in appropriate biohazard containers. Another aspect of biological safety is the use of gloves, gowns, and face protection. Consistent and thorough hand washing. So hand washing is especially important. Even though you are wearing gloves, there are certain particles that can go through the gloves and get directly on your hands, especially in the microbiology area. But when you're dealing with blood specimens, it's always good practice to consistently wash your hands several times throughout the day, especially before and after breaks or meals. Capping of specimens during centrifugation and use of centrifugation with sealed cups. So making sure those tubes are completely sealed before you put them into a centrifuge. So some more on biological safety. Bloodborne pathogens. There's a written exposure control plan that must be available to employees. And universal precautions, as I described previously. There should be a universal precautions policy, and it should be followed. For airborne pathogens like tuberculosis, there should be a tuberculosis exposure plan. TB isolation areas with ventilation controls must be established. Workers in high-risk areas must wear appropriate respirators. And airborne pathogens other than TB including some type of fungal spores and that type of stuff should be worked with in an appropriate biosafety cabinet and area. Typically with Mycobacterium, there is a separate room. It's classified at a little bit higher of a level, so BSL-3 instead of a typical BSL-2, and you have to be completely protected, usually wearing a bodysuit and some type of advanced respirator before you go into that room to start doing manipulations of Mycobacterium. Shipping. So infectious substances and diagnostic specimens must be labeled and packaged as required. The DOT guidelines are found in the Code of Federal Regulations, Title 49, Subchapter C. IATA publishes its own manual on dangerous goods regulations. For chemical safety, the Hazard Communication Standard applies. Safety data sheets, or SDS, are the major source of safety information regarding a specific hazardous material provided by the manufacturer or developed by the employer. It must, by law, include information on the health hazard, handling, storage, protective equipment needed, first aid procedures, etc. Toxic effects from the hazardous substances, toxic vapors from chemical solvents, mercury must be avoided, and that's where a fume hood comes into play. Air sampling, routine monitoring, and spill kits are required. Storage and handling of chemicals. Flammable or combustible chemicals, corrosive chemicals, reactive chemicals, carcinogenic chemicals, and chemical spills. So chemicals should be stored according to their group. And different chemicals are assigned different groups, and that group is typically on the manufacturer's label of that chemical. A lot of times, the chemicals, once they're stored by groups, will be ordered in alphabetical order within their group. Chemical spills is another thing to consider. So there's chemical spill kits and such that are available, and then appropriate chemical disposal after a chemical spill is also a consideration. Radiation safety. So caution signs and restricted access to areas with radioactive material, regular monitoring and decontamination of equipment, maintenance of records and appropriate licenses, proper training for work with radioisotopes, monitoring of employee exposure, and engineered shielding and personal protective equipment is necessary. Fire safety. A fire is a chemical reaction involving rapid oxidation of combustible material or fuel, liberating heat and light. There's 4 factors to consider with a fire: fuel, ignition source, oxygen, and reaction. One of the best ways to put out a fire is to eliminate the oxygen from the area, and we can do that using different types of fire extinguishers or fire blankets and other types of ways to eliminate oxygen exposure to keep the fire or prevent the fire from going. Classification of fires. A Class A is an ordinary combustible solid material, so like paper or wood. Class B, flammable liquids and gases and petroleum products. Class C, energized electrical equipment. And Class D, combustive or reactive metals, so magnesium, sodium, potassium, all those types of things. Types and applications of fire extinguishers. So a Class A fire extinguisher uses pressurized water, foam, and its multi-purpose dry chemical. Classes B and C are multi-purpose dry chemical, carbon dioxide, halogenated hydrocarbon for computers. Class D is a dry chemical extinguisher, and trained firefighters are only allowed to use that. Fire drills conducted regularly, and evacuation of all personnel in the event of a fire. There should be an evacuation plan, and that should be communicated to each employee. Control of other hazards. So electrical hazards, use only explosion-proof and properly grounded equipment. Check for frayed electrical cords. Promptly report malfunctions. Know the location of the electrical panel. And never operate on live electrical equipment or with wet hands. Electrical hazards may seem like they're not going to occur in clinical chemistry, but you are using analyzers, and those analyzers are connected to electricity, and you are adding wet reagents oftentimes, and solvents and buffers and such to the clinical chemistry analyzers. So electrical hazards are pretty commonly encountered. Compressed gases hazards, so store tanks vertically and always keep the cylinders secured. The securing of cylinders is a law. It's required by OSHA. It's federally regulated. There's specific anchors that will anchor the cylinder to either a desktop or a wall. Never store flammable liquids and compressed gases together. Do not force frozen cylinder valve. Cryogenic materials. Use only containers designed to withstand ultra-low temperatures. Use ion phase protection and impermeable loose-fitting gloves. Store cryogenic fluids in well-insulated but loosely stoppered containers. Mechanical hazards, balance centrifuges. So balance centrifuges we will talk about when we go through centrifugation and pre-analytical procedures, but a centrifuge spins at a very fast speed, so you have to have a balance to it. So if you put a tube in one side of the centrifuge, you have to have a counter tube on the opposite side of that centrifuge, and it has to be relatively the same weight so that when it does spin, it's going to balance out the centrifuge. If you don't do that, it could cause the bucket inside to be uneven, wobble back and forth, and it can completely damage the centrifuge. Take special care with sharps and glassware. And then there's the ergonomic hazards, and that comes with working in any type of job. You want to avoid prolonged repetitive motions, and you always want to lift with the legs. Disposal of hazardous materials. So chemical waste, you always want to neutralize strong acids or bases before their disposal. Never dispose of foul-smelling or potentially reactive chemicals down the drain. Collect designated liquid wastes, i.e. flammable solvents, in appropriate containers, and segregate into compatible classes. So you don't want to just have one chemical disposal bin for all types of chemicals. They need to be divided by chemical class. Filter and redistill solvents such as xylene and acetone for reuse. Burn flammable material in specifically designated incinerators. Transform explosive substances into less hazardous forms. Bury wastes unsuitable for incineration in designated landfills. Radioactive wastes, you should consult a radiation safety officer about policies dealing with radioactive waste disposal. Transfer radioactive materials to licensed receiver for disposal. Biohazardous waste, establish and implement an infectious waste program. Place into a bag marked with a biohazard symbol, so they're typically red in color, orange in color, and they have some arrows, and you can see the typical biohazard symbol. And then into a leak-proof, puncture-resistant container with a tight-fitting lid. Place sharps into a special puncture-resistant container, and never transport, recap, bend, or break by hand needles. Accident documentation and investigation. Report any accidents involving personal injuries to the supervisor immediately, and then maintain records of occupational injuries or illnesses for the length of the employment plus 30 years. Complete and forward log and summary of occupational injuries and illnesses to the U.S. Department of Labor, and investigate causes of all accidents.